Hello and welcome to this recap of Unit 3, Coronavirus Edition. Unit 3 is all about cellular energetics, and at the center of this is a little molecule known as ATP. Now, hopefully you remember that in order to make ATP, which is the energy currency, that's what drives a lot of chemical reactions in the cell, we need to build it from precursors, and the precursors are ADP, adenosine diphosphate, and a phosphate group. So basically we need to take ADP and phosphate and combine them together into ATP. We have a problem though, and the problem is doing this requires the input of energy. In other words, it has a positive delta G. The change in free energy is positive. And so the key to this whole unit is we need to use what we call an exergonic process, or one that releases energy, and use it to drive an endergonic process, or one that requires energy. So the production of ATP is endergonic, and so we need to combine that with an exergonic reaction. To give you an analogy for what I mean, imagine we have a teeter-totter here, and we have a person standing on one end of the teeter-totter, and our goal is to launch them up into the air. Well, to get them to jump into the air requires the input of energy, right? So to go up this way, that has a positive delta G. It is endergonic. But what we can do is we can combine that with another reaction. In this case, we'll take this heavy weight here, 100 pounds, no, it has to be bigger than that, 1,000 pounds. And we can drop that heavy weight down here. This reaction is exergonic, meaning it's a negative delta G. It's going to give off energy because gravity is going to cause this to fall down. So essentially the weight dropping is exergonic, and that will drive the endergonic launching of that person into the air. So in the case, this is called energy coupling, by the way. We are coupling an endergonic process with an exergonic process. So in order to apply this to this current unit, we need an exergonic process to drive this. If this has a positive delta G, we need to combine it with something with a negative delta G. And that process with a negative delta G is cellular respiration. So C6H12O6 plus 6O2 yields 6 CO2 plus 6H2O. So we take this exergonic process and we couple it to this endergonic process, and we can drive the production of ATP just like we can launch someone into the air by dropping a heavy weight on the end of the teeter-totter. So the rest of the unit then is just details. So how is it that we break glucose down into carbon dioxide and water to release energy? Well, there's three main processes. There's glycolysis, there's the Krebs cycle, and then there's the electron transport chain. So there's a handout that you probably want to get out, and I'm going to pause here and we're going to switch to a screencast so I can show you what those are. So you have this handout likely from class already, but if not, go to Schoology. In the Unit 3 folder, click on Study Guides, and you should find all the study guide outlines as we go through. And then at the end of those, there are some important diagrams that kind of give an overview of cellular respiration. So we're going to kind of alternate between me explaining on the whiteboard and then going into detail with these pictures. So you definitely want to pull this out and have it handy as we proceed. So this overall equation, glucose plus oxygen yields CO2 and water, can be broken down into three main phases: glycolysis, the Krebs cycle, the electron transport chain. We're going to start with glycolysis. And in glycolysis, what happens is we start with this glucose molecule. We start with glucose, which is a 6-carbon molecule. And what we do is we carry out a series of chemical reactions where we slowly break glucose down. And at the end of these chemical reactions, we end up with 2 molecules of pyruvate. Now, each pyruvate is 3 carbons, so we essentially are breaking a 6-carbon glucose unit into 2 3-carbon pyruvate molecules. This process, because we're ripping apart a molecule, is exergonic. It has a negative delta G, and that negative delta G is coupled with a couple other endergonic reactions. The first endergonic reaction it's coupled with is we can take a little ADP and phosphate and combine them together into ATP. And this is done through what's called substrate level phosphorylation. In other words, there's particular reactions in this process that are able to build that glucose or that ATP up just in one single chemical reaction. The net gain is 2 ATP. The other endergonic process is we can take something called NAD plus and convert it into NADH. In other words, we have coupled the exergonic breakdown of glucose to 2 molecules of pyruvate to the endergonic production of NADH and ATP. So now let's look at your handout and sort of zoom in on the details of this process and just confirm that I'm not lying to you. So we just got done saying glycolysis is a conversion of glucose, a 6-carbon unit, to 2 molecules of pyruvate. And lo and behold, here is a zoomed in diagram of glycolysis. And you can see that, oh gosh, there it is, glucose. And if you count those carbons, you will find there are 6 carbons there. And through a series of chemical reactions, it eventually gives rise to 2 molecules of pyruvate. Every chemical reaction here is catalyzed by an enzyme. The enzymes are listed here. And note that after an initial investment of 2 ATP, you end up getting ATP and NADH out of the deal. So reminder on how to account for these numbers is that glucose in this early step here is broken in half. And so everything from this step onward happens twice, once for this 3-carbon unit and once for this 3-carbon unit. And so you can see that in this step, there are actually 2 NADHs that are generated per molecule of glucose. And then there's 2 ATP here and 2 ATP there. And so the net gain is 2 NADH and 2 ATP during glycolysis. Okay, so once the pyruvate is produced by glycolysis, it is then going to enter the mitochondria. You may recall the mitochondria has an outer mitochondrial membrane. It has an inner mitochondrial membrane. The inner mitochondrial membrane is highly folded. Each little infolding is called, they're each called cristae, and that is there to increase the surface area for reasons that we'll discuss in a moment. So the pyruvate is going to enter the mitochondria, and then there's a little what's called transition reaction that happens. That pyruvate, those 2 3-carbon units, are converted to something called acetyl coenzyme A. And since there's 2 pyruvate, well, that's going to lead to 2 acetyl coenzyme A molecules. Each of these are only 2 carbons, though. So we started with a 3-carbon molecule, we're ending with 2 carbon molecules. What happened to the other carbon molecule? Well, the extra carbon that's lost is actually exhaled as CO2. And again, since there's 2 pyruvate converting to 2 acetyl-CoA, we get 2 carbon dioxides out of there. Now, this is also exergonic, and that drives the production of a little bit more NADH. Okay? Now, once we have acetyl coenzyme A, now it's time to enter the Krebs cycle. And what happens in the Krebs cycle is there is a 4-carbon unit. It's called oxaloacetate. I don't really care that you learn the name of that molecule, but that's what it's called if you're interested. That acetyl coenzyme A combines with that 4-carbon molecule to form a new molecule called citrate. And this is why sometimes this is called the citric acid cycle. Citrate is actually 6 carbons long. And so we can actually, if we want to do this, double everything to kind of keep our numbers straight, we can do that. Now the reason why this is called the Krebs cycle or the citric acid cycle, sometimes it's called the tricarboxylic acid cycle. The reason why we call it a cycle is because a series of chemical reactions happen that ultimately regenerate this 4-carbon molecule. So 2 carbons come in, but then along the way, we actually lose 2 molecules of CO2 per turn of the cycle. And since we start with two 2-carbon units, each turn of the cycle we lose 2 more, so then the second one comes in, and so we have to multiply this by 2 again to keep our numbers. So these 2 carbons come in, are lost as CO2, but this is happening twice per molecule of glucose, and so we have to multiply this by 2 as well. So 2 carbons come in, we regenerate our 4-carbon unit. 2 more carbons come in, we regenerate our 4-carbon unit, and so it's a cycle. What's the point? Well, along the way, we get a little bit more stuff going on. We get more NADH, and we get more ATP. So the continued exergonic breakdown of these molecules are coupled with the androgonic production of ATP and NADH. Interesting little side note, you'll note that we started this whole process with 6 carbons in glucose. By the time we're done, we lost 2 in that transition reaction, and then 4 in the Krebs cycle. All 6 carbons have now been exhaled as carbon dioxide, which explains why 6 carbon dioxide molecules are the product. Let's take a look at the technical diagram and again, make sure this is all holding up. So during the Krebs cycle, it begins with this transition reaction. Pyruvate is actually transported into the mitochondria. That pyruvate is converted into acetyl coenzyme A. If you count, you'll notice there's 1, 2, 3 carbons in pyruvate, but only 2 carbons in acetyl-CoA, and so one of these 3 carbons is lost as CO2. That process is exergonic and ends up giving off an NADH, but remember, this is happening twice per molecule of glucose, so you actually get 2 NADH there. This acetyl-CoA then enters the Krebs cycle. The 2-carbon acetyl-CoA is joined with this 4-carbon oxaloacetate to make the 6-carbon citrate. And then a bunch of chemical reactions happen, and you could kind of follow it along if you'd like, but the key idea here is that the breakdown of that 6-carbon citrate is exergonic, and that drives the androgonic production of NADH. Again, there's 2 here because everything's happening twice per glucose. 2 more NADH here. There's 2 more NADH here. And there's 2 FADH2 here, which I forgot to mention on the whiteboard. You also get 2 ATP. Along the way, you actually do lose 2 carbon atoms. So 1 CO2 is lost here, another CO2 is lost there, and those are ultimately exhaled as we breathe. So basically, 2 carbons come in as acetyl-CoA, 2 carbons come out as carbon dioxide. At the end of it, we regenerate that 4-carbon oxaloacetate, so the whole thing can happen again. That's why it's a cycle. And then this exergonic breakdown of the acetyl-CoA is used to drive the androgonic production of NADH, ATP, and FADH2. Note once again, every reaction is driven by an enzyme, and those enzymes' names are there in case you're curious.